again. Thank you for your attention, dear colleagues. I would like to present the results of a scientific project titled Genetic Polymorphisms Affecting Doxorubicin Cytotoxicity. This project was conducted at the Department of Clinical Pharmacology in the University Hospital of Göttingen, Germany. As you know, doxorubicin is one of the historically oldest chemotherapeutics, which is used in a broad range of cancer types, including breast cancer, ovarian, gastric, urinary cancer, non-Hodgkin lymphoma, multiple myeloma, neuroblastoma, lung cancer, thyroid cancer, and so on. However, we know that besides of its cytostatic action, doxorubicin also shows off-target effects, which the most concerning is an irreversible cardiac disease induced by anthracycline or doxorubicin medication. We know that nowadays the patient population ages overall, which has us to experience patients with increased age and comorbidities at the time of their cancer diagnosis. Actually, some authors found that about 40% of the overall population in the age group of between 40 to 59 years have already developed an underlying cardiovascular disease. So this is why it is very crucial and important not only to identify patients at risk by cardiologic screening prior to the medication, but also to pinpoint patients who might more benefit from doxorubicin treatment, because still showed benefit in the treatment and in the control of cancer disease. So I would like to talk somewhat about the mechanism of action of doxorubicin. We know that the primary mechanism of action of doxorubicin is related to the topology of DNA molecules and reactive oxygen species. So doxorubicin can modify the DNA topology by two ways. One would be the intercalation of the doxorubicin molecule in the DNA, which pushes apart the flanking bases, then preventing other proteins to dock onto the molecule, so mostly preventing proteins having a role in DNA replication, transcription, and even DNA repair. Then we know that doxorubicin can trap the topoisomerase enzymes at the cleavage sites of the DNA molecule, preventing the molecule to be resealed. Then the other mechanism of action of doxorubicin is related to reactive oxygen species, where due to its quinone structure, doxorubicin can be easily oxidized to a semiquinone radical by NADPH oxidase enzyme, which then is unstable and reacts further with oxygen, forming superoxide radicals and further hydroxyl radicals, which are also unstable, attack other biomolecules, causing, for instance, lipid peroxidation, or again, DNA damage at the level of nucleus or mitochondria. we opted to, by examining the mechanism of action of doxorubicin, to identify genetic markers predicting the response to this drug, so that we can fully benefit of its action and not only the side effects. So what we did, we used lymphoblastoid cell lines as model cell lines. Why? Because sorry, I wanted also to describe the candidate gene approach we used. So we were evaluating the role of candidate genes in 4 categories. So the first one in blue in the upper left would be topoisomerase isoforms. Then also we were concerning the DNA repair genes showed in red, genes related to structural and functional components of the NADPH oxidase enzyme complex shown in yellow, and representative antioxidant enzymes which were found to be expressed in our model cell line. So as a model cell line, we used the lymphoblastoid cell lines, which have the advantage of having a short doubling half time, are easy to culture, but also there is an online database of the genome of these cells. So after incubation with doxorubicin for 48 hours, we measured functional parameters which included the vitality, or the EC50, which was defined as the concentration at which the cell vitality would drop to 50% of the maximum effect. Then we would measure the production of reactive oxygen species by using DHCF as a proxy for that. And we would measure doxorubicin internalization, which was quite easily done by effects, given the fluorescent nature of the compound. Then also on the same cell lines, we measured PMA-induced activity of the NADPH oxidase enzyme complex by measuring luminescence. So then to our results, we observed a strong negative correlation between the formation of reactive oxygen species upon doxorubicin incubation and the EC50 values. Meaning cell lines with low EC50s exhibited more reactive oxygen species formation. However, well, this could mean that ROS are the main mechanism of action for the doxorubicin cytotoxicity. However, then we would expect a more sinusoid curve here, because by this increased cytotoxicity of the cells, actually at the end, the reactive oxygen species formation would decrease as the cells would be dying, which we didn't see here. Then next slide, I can show you we observed a positive trend between the doxorubicin internalization and the PMA-stimulated NADPH oxidase activity, which shows that doxorubicin can affect cell vitality also indirectly by activating reactive oxygen species producing enzymes. Then also we show the correlation of doxorubicin internalization and reactive oxygen species production after incubation with doxorubicin. So as we see, reactive oxygen species play a major role, but are not the only ones. Then we focused on the evaluation of the impact of genetic variants of the mentioned candidate genes on the doxorubicin cytotoxicity. And out of all candidate genes and variants examined, only one, which was found in the topoisomerase 3A, was significantly associated with a higher sensitivity towards doxorubicin in both the training set and the test set. This polymorphism is located in the second last intron of the gene coding for this enzyme and increased the cytotoxicity of doxorubicin by about 30%, which is quite significant. Interestingly, the frequency of the variant allele counts for about is about 9.5% in the Caucasian population, so could be important in clinical practice. Next, showing a table of SNPs in DNA damage and repair machinery. Here, we only found SNPs which were significant in the training set, but the results couldn't be seen superimposed on the test set. But the most significant variants were identified in a gene coding for protein 53. We found no association with the constituents of NADPH oxidase enzyme complex. And also similarly, no significance was found in the genes coding for the antioxidant enzymes catalase and superoxide dismutase 1. So to finalize the results, we found that reactive oxygen species formation was the major route for cytotoxicity of doxorubicin. And our results indicate that genetic variation in the topoisomerase 3A gene may serve as an important predictor of the therapeutic response towards this drug. Controversially, we did not find any significant impact of the SNPs in candidate genes encoding other topoisomerase enzymes, the DNA repair proteins, genes related to NADPH oxidase complex or antioxidant enzymes. As it always is, to validate these results, we need further case control approach and also maybe evaluation not just in vitro cell cultures, but in patients. But also, I suspect that the mechanism driving the cytotoxicity of doxorubicin is of polygenic nature. So reactive oxygen species have a big impact, but the SNPs were actually shown in the topoisomerase coding genes. So I would like to address the role of topoisomerase 3A a bit. It was found that this enzyme is expressed in a wide range of tissues, so it's relevant. However, it is exceptionally high in endothelial cells and stromal fibroblasts, which is interesting regarding the cardiotoxicity of doxorubicin or the cardiovascular, actually the vascular part of the diseases it causes. And interestingly, topoisomerase 3 alpha is one of the isoforms present in both the nucleus and mitochondria. And its role is to maintain the genome stability at both sites, although mutations in the enzymes were reported in mitochondrial disorders mostly. One of them, as mentioned by previous speaker, the Bloom syndrome, which is where topoisomerase 3A is related to telomerase activity. Several authors have discussed the differentiated expression of topoisomerase isoforms as a source of adverse effects of doxorubicin. It was postulated that the drug inhibits mainly isoform located it was postulated that the drugs mainly inhibit the topoisomerase 2A isoforms in cancer tissue, and controversially topoisomerase 2B in cardiomyocyte. That's why the cardiovascular side effects are so pronounced and other tissue stays unaffected. The 2B isoform is mainly located in mitochondria, similarly to the 3A isoform, of which cardiomyocytes are particularly abundant due to their function and metabolism. The impact of the 3A isoform in the context of doxorubicin has not been addressed so far, and currently there is no supportive medication selectively protecting mitochondria from the side effects of the chemotherapy. But maybe the answer lies in the mitochondria, and if we could protect the mitochondria in the future and have a selective effects more directed towards the nuclear DNA, maybe we would have the best solution and the best of words in the doxorubicin treatment. So I would like to thank you for your attention, and also to thank to the co-author of the project and the supervisor in the lab, Marcus Schirmer, is currently working at the Department of Radiotherapy and Radiation Oncology at the same university on Göttingen in Germany. And also say thank you for the funding, which was given partly by the Ministry of Education of Slovak Republic, where I come from, and the German Research Foundation. Thank you.